What if I told you that Frankenstein didn't just come from a writer's imagination? That there were scientists who actually tried to bring the dead back to life, and that a simple thunderstorm changed the history of biology? Today we're going to look beyond the myth, beyond the movies, and beyond Frankenstein himself. Today we're going to look at the real science that inspired the idea of creating artificial life, and what you're about to discover will send shivers down your spine. Welcome to Beyond DNA. I'm Claudia, and today we're going to tackle the question that obsessed Galvani, Aldini, and Mary Shelley. To what extent is life an electrical phenomenon? Before Mary Shelley invented the monster, there were already scientists attaching electrodes to dissected animals and human bodies to see if electricity could make them move again. Many people left those experiments convinced they had seen the boundary between life and death tremble on a stretcher. Through Frankenstein, we're going to learn what bioelectricity is and how it works in your brain, your muscles, and your heart, and how those bizarre experiments ended up being the origin of a part of modern biology. Let's set the scene. Summer of 1816. A brutal storm. A group of young writers locked away in a mansion by Lake Geneva, and one of the most famous literary challenges in history to see who could write the best horror story. Among them was Mary Shelley, just 18 years old. And while the wind battered the windows and thunder lit up the sky, Mary began to hear stories about real experiments. Scientists who tried to reanimate dead animals using electricity. The seed of Frankenstein didn't just come from imagination, it came from the science of the time. If you've seen Guillermo del Toro's film, you already know that the monster isn't just a monster. It's a reflection on life, death, and the responsibility of the creator. But to truly understand the origin, we have to travel to the laboratory where this idea was born. Let's meet the man who started it all, Luigi Galvani, an Italian physician from the 18th century. One day, while dissecting a frog on a table with an electrostatic machine on it, something happened that changed his life. The leg of the dead frog moved on its own when a metal scalpel brushed against its nerve. Imagine the shock. A corpse moving as if it were alive. Galvani repeated the experiment until he reached a revolutionary conclusion. The body holds animal electricity, an internal force that travels through the nerves and causes muscles to contract. What he called animal electricity is what we now know as the electrical behavior of cell membranes. Each neuron maintains a difference in charge between the inside and the outside. Inside, it's more negative, and outside, more positive. This voltage difference is maintained by proteins like the sodium-potassium pump, which moves sodium out and brings potassium in, as well as ion channels that allow some ions to pass more easily than others. The result is a resting membrane potential. The cell is calm but ready to fire. When a strong enough stimulus arrives, voltage-gated ion channels open. Sodium enters, the membrane depolarizes, and a threshold is reached. An action potential is triggered. A voltage spike travels down the axon like a wave, opening and closing channels as it goes. This impulse reaches the synapse. There, calcium enters and neurotransmitters are released. At the neuromuscular junction, the star neurotransmitter is acetylcholine. It binds to its receptors on the muscle fiber, channels open, a new action potential is generated in the muscle, and calcium is released from the sarcoplasmic reticulum. Calcium binds to the contractile proteins, and the actin and myosin fibers slide past each other. The muscle shortens, and as a result, movement occurs. What Galvani saw in his frog was a simplified version of this entire circuit. The external current acted as a nervous muscle, depolarized the nerve, triggered action potentials, and the muscle contracted. Of course, for 18th century science, this was a shock. Until then, it was believed that life was something mystical, a divine breath, not a combination of ions, gradients, and chemical signals. And if electricity moves muscles, the inevitable question arose, could electricity bring life back? And this is where everything gets very Frankenstein-like. Giovanni Aldini, Galvani's nephew, decided to show the world that his uncle was right. And he did it in a big way. In 1803, in London, he obtained the body of a criminal named George Foster who had just been executed by hanging. And in front of doctors, journalists, and onlookers, he connected electrodes to the corpse. What happened is documented and real. When Aldini applied electric current to the face, the corpse's mouth twisted into a grimace, the eyes opened, and the head moved as if trying to breathe. When he applied electricity to the hand, the fingers closed into a fist, and when he stimulated the legs, the entire body went into violent spasms as if trying to get up. For the audience, it was like watching a dead man come back to life. Obviously, there was no consciousness or functioning brain. They were just muscle contractions caused by electricity. But imagine hearing stories like that at 18, in the middle of a storm, in a remote mansion. What story would you write? Mary Shelley wrote Frankenstein. And that's where cultural history diverges from scientific history. But biology kept going. What Galvani and Aldini were tapping into without knowing it was the foundation of how your nervous system works. What they experienced as something almost magical, we can now describe with ions, channels, and action potentials. So far, we've talked about neurons and muscles, but bioelectricity goes beyond the nervous system. Think about the heart. The heart has its own electrical system. In the sinoatrial node, there are specialized cells that depolarize rhythmically. They are the natural pacemaker. Every time they depolarize, they send out a signal that spreads through the atria, passes through the atrioventricular node, travels down the conduction pathways, and reaches the ventricles. It's like an electrical choreography. Each cell depolarizes and repolarizes in order. And thanks precisely to this, the contraction is effective. Blood comes in, blood goes out, and circulation is maintained. When that system breaks down and, for example, ventricular fibrillation occurs, the heart cells still have action potentials, but in a chaotic way. There is no longer any order. The heart quivers, but it no longer pumps. It's a critical situation, and once again, the tool we use is bioelectricity. A defibrillator doesn't start a stopped heart as if it were a car with a dead battery. Instead, it delivers a strong, very brief shock to massively depolarize the tissue. If enough tissue is still alive, the natural pacemaker can resume setting the rhythm. If not, the heart does not recover. We're not bringing someone back to life in the Frankenstein sense. We're intervening in an electrical system that still has the potential to reorganize itself. Up to this point, we've seen bioelectricity as a signal, impulses traveling, muscles contracting, hearts beating. But in recent years, another idea has emerged: bioelectricity as a blueprint. Not just as a fast signal, but as a map that helps determine how the body is built and repaired. Different groups have observed that the cells within a tissue do not all have the same membrane potential. In an embryo, a regenerating limb, or a healing wound, voltage patterns appear, areas that are more depolarized and areas that are more hyperpolarized. An electrical pattern along which cells move, divide, and organize themselves. If you alter these patterns with drugs that activate or block ion channels, you completely change the behavior of the tissue. In models like tadpoles or regenerating animals, it has been observed that you can promote limb regeneration, induce the formation of structures outside their usual location, or partially correct malformations by altering the electrical signals that the cells receive. Importantly, without touching the DNA, just by changing the bioelectric context. The genome is still the foundation, it's the list of parts, but the way these parts are arranged also depends on electrical signals. Bioelectricity works as an extra layer of information, like software that says, here goes a leg, here goes an eye, here this wound needs to close, or here growth needs to stop. If we understand this software, we can think of very different applications. Regenerating tissues, improving healing, preventing certain cells from entering a tumoral mode, designing organs in laboratories that organize themselves in a more orderly way. And yes, it also opens the door to something that sounds very Frankenstein-like. Guiding the behavior of living structures we've created in the lab. Today, we're not stitching together corpses. What we're making are organoids, mini organs generated from stem cells, mini brains that produce spontaneous electrical patterns, mini hearts that beat in sync on a plate. In all these areas, bioelectricity appears on its own because it's part of how life works. But it can also be modulated from the outside. We could record those patterns with electrodes, apply electric fields, combine electrical signals with chemical and mechanical signals. And we're getting closer and closer to understanding how to use this to design functional tissues. And this is where Frankenstein stops being just a scary story and becomes a metaphor for something very current. The novel isn't just about a monster, it's about a scientist who crosses the line without thinking about the consequences. Today, instead of a mysterious spark in an attic, we have gene editing with CRISPR, organs on chips, organoids in the lab, deep brain stimulation, pacemakers, defibrillators, and a biology of regeneration and artificial life that's advancing very quickly. The important part is to understand that behind all these techniques lies the same foundation discovered by Galvani and Aldini. Cells are electrical. Life, as we know it, depends on voltage differences and microscopic currents. That gives us power. We can intervene in failing hearts, in neurons that don't communicate properly, and in tissues that don't repair themselves. But it also puts us in the uncomfortable position of Victor Frankenstein. We now have the real ability to touch the basic mechanisms of life, and we have to decide how, when, and to what extent we want to do so. Galvani saw a frog's leg move and thought of a new way to understand life. Aldini saw a corpse convulse and believed he was on the verge of resurrection. Mary Shelley saw those experiments and wrote a warning disguised as a novel. We, 2 centuries later, already know that we are not going to bring the dead back to life with a lightning bolt, but we are learning to read and edit the electrical code that keeps an organism alive. And the real question is no longer whether life is an electrical phenomenon. That much is quite clear to us. The question is no longer, can we? The real question is, should we? Frankenstein was written more than 200 years ago, but it is still completely relevant today. It's not about the fear of science, it's about the fear of using science irresponsibly. If you've made it this far, congratulations. You've just survived a biology, history, and electricity lesson worthy of Dr. Frankenstein himself. Let me know what you think in the comments. Do you think that one day science will be able to create artificial life? Or do you think we've already created it without realizing it? See you next week with another topic that seems paranormal, but is actually pure biology. This is Beyond DNA, and today you've looked inside the lightning that gave life to the monster.